Good evening, good evening. Am I audible? सुनाई दे रहा है? Okay, we'll wait for some time, then we'll start the class. This is the last free class on YouTube for plant physiology or crop physiology. After that, we will start on the app itself, on the paid classes. Uday Lakshmi is very punctual. They always come on time. That's good. You should be punctual to crack any competitive examination or to do anything in life. The first step is discipline, and under discipline, the punctuality comes. Okay. Welcome, Manikegam. Definitely will enjoy the class. The class will be more conceptual than factual, and you will understand the reason behind the concept and the why of the study. Okay, the why behind the topic you will understand in my class. So even if you haven't understand anything in UG classes, you will be able to understand here. So don't worry. And if even if after that any doubt you have, then you can ask me on WhatsApp. I will reply there. And on paid class also, we will be interacting. In paid classes, the classes will be interactive classes. We both will interact and then discuss there only, no doubt. Okay, so let's start. So as today's topic was on, like now topic was on osmosis, diffusion and imbibition. Okay, so basically the substance get exchanged between different mediums or different ecosystem, different system. The substance are exchanged between system. So how this substance get exchanged between the systems? There are basically three process. And imbibition actually, it is not a process of exchange, but it is the process of absorption. But diffusion and osmosis, they exchange. How the things are exchanged? Through diffusion, gases are exchanged. Diffusion में gas exchange होते हैं. And in osmosis, the liquid or solvent but move from one system to another system, right? And in imbibition, what happens? In imbibition, solid surface here. Here is a solid surface. Okay, like in cell shape, there is protein, the colloidal particles, they are also solid. So on the surface, the water get absorbed. Okay, only the surface part. It will not enter or bypass the system. That is imbibition. The swelling of wood, you can say it is a process of imbibition. Okay. So first we will discuss about diffusion. What is diffusion? See, the substance enter and leave the plant cell in solution from dissolved in water or as gases. Exam there are three main physical process are involved: the diffusion, osmosis, and imbibition. So first we will see diffusion, then we will see osmosis, then we will go for imbibition. Okay, so what is diffusion? Diffusion, the exact meaning or the normal literal meaning of diffusion, can anyone say? I'm not asking the definition according to plant physiology. The normal meaning of diffusion. Consider diffusion as an English word, and what is the meaning of diffusion? Uday Lakshmi, the definition is correct, but it is not the literal meaning. It is the definition from plant physiology or scientific point of view, science point of view, right? So literal meaning will diffuse means moving from one place to another place. The diffusing, the people are diffusing or anything can diffuse means the movement from one place to another place is the diffusion. So in plant physiology, the movement of particles or molecules, anything, the molecules or particles or elements, they move from one place to another place, but they follow a path, definite path or a definite direction. And who gave the direction? Okay. So for example, this is one system, this is one system. Okay. So here are some particles, and here also are some particles. And the direction is this. But what decided this direction? What is the thing that decided this direction and what this direction is called? Okay, so the particles will move from here to here. So it is high concentration of particles or molecules, and there is low concentration, right? So the diffusion takes place along the concentration gradient, means from higher concentration to low concentration. And gradient is nothing but gradient means slope only. Gradient is slope. Slope is like this, no? Here it is slope. So from higher to lower, this is the direction of flow. So in diffusion, the molecules move from one system to another system on the basis of the gradient or slope, that is from higher concentration or higher gradient to lower gradient, right? Lower concentration until they are evenly spread. And how long this diffusion will continue? Until the two systems come into equilibrium. Equilibrium means the concentration of system A is equal to concentration of system B. So when this both system came into equilibrium, only then the diffusion process will stop. Okay. So what is diffusion pressure now? Like presence of something at any place will create any pressure. Like if I am keeping this pen here, it has some mass, it will definitely create some pressure on my palm, right? So presence of anything will create a pressure, right? So here we saw system A was on high concentration. High concentration means the number of molecules are more, right? Say for example, the number here it is CO2. CO2 molecules are there. Okay, same molecules, but number of molecules is much more. बहुत ज़्यादा molecules हैं यहाँ पे है कम. So more the molecules, more will be the mass, right? And more will be the mass, greater will be the pressure. Why? What is the formula of pressure? Pressure is equal to per force per unit area, right? And force is equal to mass into acceleration, or you can say force is directly proportional to mass. So greater the force, if mass will be greater, force will be greater, and if force is greater, pressure will be greater. So in higher concentration, the pressure will be more. So if the number of molecules in a system is more, it has more diffusion pressure. Understood? Diffusion pressure, the particles have natural tendency to move. This is called diffusion pressure. The natural tendency due to the difference in concentration or the number of molecules, the molecule has a natural tendency to move. And why they move? Because of difference in the diffusion pressure between the two systems. Okay. Higher concentration, higher diffusion pressure. As I explained, why higher diffusion pressure? Because more molecules will have more mass, and more mass then more force then more pressure. That's why higher pressure, higher concentration, much number of molecule, and if the number of molecules are more, they will create or they will exert greater pressure along the diffusion pressure gradient. Okay, this is the direction, right? So the diffusion always occur along the gradient means along the slope. Okay, from higher concentration to lower concentration, this is the gradient. Gradient is like a path that is from a higher point to lower point, that is gradient. And it is here we are talking about pressure, the diffusion pressure or pressure gases that exerted by gases. So here it is we taken as diffusion pressure gradient. We call it diffusion pressure gradient because it is denoting the pressure. Okay, now what are the factors that affect diffusion? Diffusion is a natural process. It will occur pressure. See, in a system, the presence of solute also creates some pressure. Pressure doesn't always external. Yes, external we are applying the pressure. But in a system, the presence of molecules or the presence of substance create a pressure itself. Right? If I am putting here a pen, am I exerting some pressure here? No, the external pressure is not exerted. But the pen itself, due to its mass, creating some pressure or exerting some pressure on my hand. Understood? Even if I will close in, it is in a like container, the pen will still exerting some pressure. So here also the pressure is due to the molecules present inside only, and the pressure is due to the mass possessed by the particles or molecules. Molecules have mass, right? Uday Lakshmi, the molecules have mass. So because of its own mass, it is exerting some pressure, and because of that pressure, the system A has high diffusion pressure because the number of molecules are more, and it has also more pressure. Okay. So now what are the factors affecting the rate of diffusion? Diffusion automatically occurs along the gradient, higher concentration to lower concentration, right? It is automatic. No external force is applied here, right? So what are the factors that affect the rate of diffusion? Rate means the amount per time. How much amount of gases are diffused along the pressure gradient per second or per minute or per hour. That is rate. So rate can be more, like more number of molecules can be diffused. Yes, concentration per time, that is amount or concentration per time. Amount only concentration, right? If the amount of solid is more, the concentration is more. Okay, so the amount of molecules or the concentration of molecules per time is the rate. The rate can be dependent on various factors. It may be more or it can be less, right? So if the concentration gradient or the difference between high concentration and low concentration is very large, it means if high concentration or the system at higher concentration of solute or molecules, suppose it is 50 atm, okay, and the low concentration part or the system B, wait, we will learn in according to system. Suppose in system A, the diffusion pressure is or the concentration is 50 atm. Okay, and in system B, the diffusion pressure is 25 atm. This is case 1. And in another case, that is case 2, here the pressure in system A is still 50 atm, but the pressure in system B is equal to 10 atm. So where the rate will be more? In case 2 or case 1? Where the rate will be more? The rate of diffusion, where it will be more? Case 2. And why in case 2 the rate will be more? Why it will be more in case 2? Because the gradient is steeper here. Why the gradient is steeper here? Because the difference between the concentration is more. Here it is 40 and here it is only 25. So the gradient, this is the gradient difference. The pressure difference is the gradient. The pressure difference between two systems is the gradient between two systems. Yes. So here the gradient is less, means it is less steeper as compared to case 2. Here the gradient is more steeper as compared to case 1. So steeper the gradient, faster will be the diffusion, and higher temperature, faster movement of particles. Why at higher temperature the particles move faster? Because temperature, it is heat, heat is energy. If you provide more energy, the velocity of particles will increase, and when the velocity of particle will increase, then it will move faster. Okay. And there are two types of diffusion in plants, basically, that is simple diffusion. Simple diffusion means only the movement of particles from higher concentration to lower concentration. That is simple diffusion. And another one is osmotic diffusion. Osmotic diffusion means, here see, here is one system is there and here another system is there. This is A and this is B. Okay. And here there we are putting on semi-permeable membrane. So the movement of molecules from one system to another system through a semi-permeable membrane is known as osmotic diffusion. Osmosis itself only called osmotic diffusion, because in osmosis also the molecules, the solvent particles are getting diffused through a semi-permeable membrane from one system to another system, right? So osmotic diffusion is nothing but osmosis only. So next we will see what is osmosis. So before we'll see osmosis, let's see what is the biological importance of diffusion in plant. The first is gas exchange. Gas exchange occur through lenticel in plant or through stomata. Stomata is well known. Stomata is well known for gases exchange. We know the carbon dioxide get exchanged through stomata only, right? So gas exchange, CO2 in photosynthesis also, stomatal, O2 out in respiration, and salt uptake through absorption through root. Root absorb the salt or a nutrient with by absorbing the water. In water, the nutrient are dissolved, and that dissolved the nutrient dissolved water is absorbed by the root of plants. That is also occur through osmosis, right? And long distance transport, movement of gases through open stomata. Okay, this is the biological importance of diffusion, like how diffusion helps in the movement of gases from one part of plant to another part of plant, or from atmosphere to plant and plant to atmosphere. Okay. Mainly diffusion help in movement of gases. And the key characteristic in diffusion, it is a vesicle summary. Like till now we have covered diffusion. So have you seen anywhere that diffusion is energy dependent? Some external energy is required to initiate a diffusion process, or it is natural, spontaneous, it occurs by its own. Right. Answer please. Diffusion is energy dependent or it is occurring by its own? Diffusion is not requiring any energy, external energy or external input of energy. Diffusion is a spontaneous process which only depend on the concentration gradient, right? Or the diffusion pressure gradient. If gradient is very steeper, it is faster, and if it is less steeper, the movement is slower. So energy required? No. That's why if the energy is not requiring, that's why the diffusion process is a passive process. And if it would have, if someone or some processes require energy, then it is an active process, right? So here diffusion is passive because it is not using or utilizing any energy or external input of energy. That's why it is a passive process. And the direction is, we know, high concentration of molecules to low concentration of molecules. Okay, membrane, there is no use of membrane, or membrane is not required in diffusion. And the speed depends upon, speed depends upon the concentration and the temperature, right? But in case of gases, liquid and solid, the diffusion of gases is much more than diffusion of liquid, and the diffusion of liquid is much more than solid. It is because the gases can flow faster than liquid. It's because of the flow speed and nothing else. Okay, now osmosis. As I said earlier also, osmosis is movement of solvent molecules. See, in solution, no? In solution, there are two things. One is solute, one is solute plus solvent, right? This is solution. So in a solution, there are two things. One is solvent and one is solute. Solute is the solid part that is dispersed in the solvent, and solvent is the water or liquid part that both after combining they form solution. Okay. So the movement of solvent molecules. Remember this: the solvent molecules means the water. Solvent is basically water in case of plant cell. Okay, water. So here the movement of water through a semi-permeable membrane from a region of low solute concentration to high solute concentration. Let's understand this. What it has told, there are two systems, okay, and these are connected through a semi-permeable membrane. This is semi-permeable membrane, okay, and here this is high solute. High solute. Please focus solute, not solvent. Here it is low solute. And this is solution A, and this is solution B. So tell me from where to where the solvent will move. Where the solvent will move. Here in solution A, the concentration of solute is high. So here the concentration of solvent definitely less if the volume is equal, right? It will be less. The solvent will move through a semi-permeable membrane from B to A. Yes, the movement of solvent would be from B to A because here concentration of solvent will be less and here concentration of solvent will be more. So from higher concentration of solvent, because solvent is moving here, solute is not moving. Semi-permeable membrane means it is only permeable to selective molecules or selected molecules. It is not permeable to solute. It is only permeable to solvent, means only water can pass through it. Water can pass through it. Solute cannot pass through it. So it only allows the solvent to pass through it. So that's why only solvent will move. So if the concentration of solute is low, then the concentration of solvent will be more. And here the concentration of solute is high. It means the concentration of solvent is less. So here the concentration of high solvent, high concentration to low concentration of solvent. So solvent will move in this direction. Understood? Okay. Here low solute concentration to high solute concentration it will move. And it is also called osmotic diffusion. I told you in the earlier slide. And key condition, membrane must be semi-permeable. Semi-permeable means only it allows the solvent to pass through it and it will not allow the solute or blocks the solute. Okay. Now what is osmotic pressure? Okay, before starting this, I would like to tell that this is the old terminology or old thing. In the modern area or in modern times, there is a different concept. So after this, we will discuss also the modern thing, the what is considered now. अभी क्या माना जाता है, वो भी हम discuss करेंगे after this. Okay. So pressure developed due to solutes means here it is a solution like solvent is there. ठीक है, solvent है, solvent में हमने कुछ solute डाला. So the amount of solute would decide what will be the osmotic pressure. Amount of solute would decide the concentration, and this concentration of solute would decide the osmotic pressure of the solution. Okay. So more will be the solute, more will be the osmotic pressure. Right. Less will be the solute, less will be the osmotic pressure. Right. So here you tell me, here it is A with high OP, high osmotic pressure. OP means osmotic pressure, and here it is B. It has low osmotic pressure, low OP. So the movement of solvent will be from where to where? From B to A. Yes, from B to A. Manikangam is right. So the solvent will move from B to A. Why? Because it is a low osmotic pressure means low solute, right? Low solute means more solvent, and here high osmotic pressure means more number of solutes. More number of solutes means less solvent. So more solvent to less solvent. It will flow from B to A. Okay. And this osmotic pressure, the unit for osmotic pressure, it is measured in atmosphere, or you can say ATM as well, ATM, and directly proportional to solute concentration. We see that more number of solutes, more osmotic pressure, right? Pure solvent has zero osmotic pressure. Why? Pure solvent has zero osmotic pressure. Pure solvent means absence of solute. Suppose here only water is present. Pure means free from impurities, right? If we add solute to this, it will get impure. It will no longer remain as pure, right? So pure water has zero osmotic pressure. This is important. Questions has been asked from this point, that what is the osmotic pressure of pure solvent or pure water? The answer will be zero. Okay. And if we will add insoluble substance to water, what would happen to osmotic pressure? Yes, money can come. Yes, yes, yes. It has no solute. That's why the solute potential is zero. Osmotic pressure is zero. If we will add insoluble solutes to a solvent, what would happen to its osmotic pressure? It will increase, decrease, or remain same, no impact. Answer, answer, please answer. Okay, let it be. So if we'll add insoluble solutes, it does not increase osmotic pressure because the solvent will be separated, and insoluble means it is not get soluble homogenized with the solvent. So it will do nothing to the solvent, right? It will not increase the concentration of solvent. It will get settled down, insoluble, then water will be pure only and it will move. So there will be no change in osmotic pressure. In a tonicity term, hypo, hyper, and iso. So let's understand three terms first. First is hypo, hyper, and iso. What is the meaning of this three term? Iso means same. Okay. Hyper means बोलते ना हाइपर टेंशन, मैं हाइपर हो गया. Hyper means more or greater. And hypo means less. Okay. So hypotonic, tonic, this word came from tonoplast. That is that is the outer membrane of vacuole, I guess, tonoplast. So hypotonic, hypertonic, and isotonic. The system. So hypotonic means hypo means less, less concentrated. Hypertonic, more concentrated. Isotonic, equal concentration. So this term has been used when there are two systems. It is a relative. These are relative terms. You cannot say single system it is hypotonic or hypertonic until and unless you have one measuring parameter, right? So these term are relative term. When two system are getting compared, or when you are comparing the two system, only then you can say that it is hypertonic as compared to system A or system B or system C, anything, and it is, or it is hypotonic or it is isotonic to that system, right? So osmotic pressure, if it is hypotonic, it's less concentrated, osmotic pressure will be low, and water movement, water moves out. So for example, in this system, okay, it is hypotonic as compared to system B. So from hypotonic means it is less solute or more solvent. So from here to here the water will move. So in case of hypotonic solution A hypotonic system, the water moves out from the system. The water moves out from the system where the osmotic pressure is low or solute is low. Okay. And hypotonic would be opposite of hypotonic, means here water will enter into the hypertonic solution. Or the solution which has more solute concentration, the water will enter into it. And if it is isotonic, there will be no net movement. Neither it will go out or neither it will come in. Hypotonic means low concentration of solute inside the cell. Yes. And more concentration of solute outside the cell. Hypertonic. Hypotonic means low concentration of solute inside the cell. Yes, Manikanam. Manikanam. And more concentration of solute outside the cell. No, it is not necessary. Okay, we are comparing two systems. And yes, you are also right. You are right. It is right only that inside the cell, the solute concentration is less, and outside the cell, the solute concentration is more. Then the cell will be called as hypotonic system. Right. So the water will move from the cell into the system. Okay. And hypertonic is reverse of hypotonic. Osmosis in plant cell. So until now we were looking only the general process, that what is the meaning of the term hypotonic, hypertonic, what is osmosis, it occurs through the semi-permeable membrane. So what is the significance of osmosis in plant cell? How osmosis occur in plant cell? There are numerous cell, right? The soils are linked to one another, they are joined to one another. We are multicellular organisms. Number of cells are present inside us. So how osmosis applies inside our body or the plant body? Okay. And the function of semi-permeable membrane is played by plasma membrane in the plant cell. Plant cell plasma membrane is semi-permeable, means it is selectively permeable. It only allows some solutes or some solvent to enter into it, and it check the outside movement, right? Same, it selectively permit the solute or the solvent to pass through it. It will not allow every or almost all solute or solvent to pass through it. Okay. If external solution is hypotonic, external solution is hypotonic. मान लो ये सेल है, and this is the external environment. This is external environment. This is external environment, and it is cell. This is cell. Okay. What it is said? The external environment is hypotonic. External is, it is hypotonic. Hypotonic means low solute as compared to cell. So definitely it will have hypertonic one. Hypertonic. So the movement of solvent will be, here it will occur exosmosis or endosmosis. Do you know what is endosmosis and exosmosis? Where the water will move? No, no exosmosis. Exosmosis will not occur. Cell is hypotonic, right? Hypotonic means more solute. and external it is hypotonic, less solute, it's more solvent, more water. Water will move from more solvent to less solvent, or low solute to high solute. B will be right. The water will move from hypotonic to hypertonic. Endosmosis will occur. Endosmosis will occur. Water enters the cell. And same, if the external source is hypertonic as compared to the cell, exosmosis will occur. Means the water will move out of the cell into the external environment. And isotonic, there will be no net movement. Okay. So pressure concept, let's see the pressure concept. At equilibrium, this is turgor pressure is equal to wall pressure. What is wall pressure and what is turgor pressure? What is turgor pressure and what is wall pressure? Well, what is turgor pressure and what is wall pressure? See, this is cell wall, right? So when water enters into the cell, it will increase the pressure that is exerted by plasma membrane or the water inside the cell on the cell wall. So the pressure exerted because of endosmosis on the cell wall is turgor pressure. Means the cell is become turgid. The pressure of the cell wall on the cell membrane to prevent from rupturing the cell. That is wall pressure. Okay. The pressure exerted by cell wall is wall pressure. And the pressure exerted by the turgor of the cell shape due to the endosmosis, that is turgor pressure. Means exerted on cell wall is turgor pressure and exerted by cell wall is wall pressure. Here it is wall pressure. And this is turgor pressure. Okay. The pressure exerted by cell shape on cell wall is turgor pressure, and wall pressure is pressure exerted by elastic cell wall against the turgor pressure. What is the significance of osmosis? Absorption by root is occurred through osmosis. Like for example, it is the soil, roots, and here the nutrients or water molecules. Generally water, the soil, so water will be here, and here it is nutrients are dissolved. So the root cells become hypertonic or high solute concentration. That's why the water from the soil moves into the root cell, and then it will move upward the plant. So in water absorption by root, osmosis helps. Okay. So if the osmosis is known, we can know the process how the root absorb water. Also cell to cell movement and other substances also move by the process of osmosis. Opening and closing of stomata depend upon the turgor pressure of the guard cell. Guard cell are like this, right? Stomata. Here it is stomatal pore. This is guard cell. Here subsidiary cell used to lie. Okay. So when water is entered into guard cell, it swell and the stomatal pore open, right? And when the water goes out, it become flaccid and the pore become closed. It become like this. So in opening and closing of stomata also osmosis help. Due to osmosis, turgidity of cell and hence the shape of the form their organs is maintained. Turgidity, it will maintain the shape of the organ also. Like because turgor pressure and wall pressure they cancel each other and maintain the shape. It is also osmosis. It is an important point. The resistance of plants to drought and frost increases with increases osmotic pressure of their cell. What is the reason? These points want to convey that the resistance of plant to drought and frost increases with the increase in osmotic pressure of the cell. Why? See here. Suppose this is cell. Okay. Suppose this is root cell. Root cell. This is root cell. Okay. So water need to enter here. For that, this system should be hypertonic. And if it will be hypertonic, then only water will enter. But in case of drought, the water, the amount of water in soil, water is less, isn't it? Then it will also become hypertonic. So how the root cell will absorb water? In drought condition, there is one mechanism occur inside the plant. What is the mechanism? Plant cells will produce certain molecules like proline. This amino acid, this chemical or this molecule will increase the solute pressure or the osmotic pressure of the cell more, and even in the drought condition, the tightly held water from the soil absorbed by plants. So it is also demonstrated through osmosis. Okay, and same in case of frost, what the higher solute concentration would decrease the freezing point of the solution. And if the freezing point will be lowered, then in case of frost also, the water inside plant will not freeze or not become ice crystals. And if you not become ice crystal, the normal function of plant will be carried out smoothly. Another concept is plasmolysis. Plasmolysis is two stage. One is incipient plasmolysis and another one is complete plasmolysis or only plasmolysis. So plasmolysis is basically a process where the protoplasm shrinks and the water inside the protoplasm, that everything goes out, or the water inside the protoplasm moves out from the cell to the hypotonic solution surrounded by it. So plasmolysis can be caused by placing the cell in a hypertonic solution. Hypertonic as compared to the cell. Okay. So plasmolysis is the process where the protoplasm sinks away from the cell wall when plant cell is placed in a hypertonic solution. Whether do you understand hypotonic, no? Hypertonic, understand or not? Okay. So you can say, what is plasmolysis? You understand already. And what is deplasmolysis? The opposite of plasmolysis is deplasmolysis. Right. And endosmosis means movement of water from outside to into the cell. And exosmosis, if water is moving out of the cell. All these terms are clear. Okay, incipient plasmolysis. Incipient means the early. The early one is known as incipient. So beginning stage is called as incipient plasmolysis. When protoplasm starts to shrink but still touches the cell wall. Okay, I will show the pictures now. See here. Here, this is the protoplasm, right? Protoplasm. And outside it is cell wall. Normal cell. Here, the protoplasm still touches the cell wall from every side. But here it look. The plasma membrane started to shrink. Right. So this stage is known as incipient plasmolysis. And this stage it is plasmolysed because it taken a spherical shape and detached from most of the cell wall. So this stage is known as plasmolysed stage or plasmolysed cell. And when it just start to shrink, it is incipient plasmolysis. The earliest stage of plasmolysis is known as incipient plasmolysis. What is the advantage of plasmolysis? It indicates the semi-permeable nature of plasma membrane. Nothing to explain here. This phenomenon is utilized in salting of meat, fishes in addition of concentrated sugar solution to jam. Right. Plasmolysis means when water is moving out of the cell. Have you noticed that in pickle preparation or in jam or jelly preparation, we add a lot of sugar or solute and we make the jam or the pickle hypertonic. Water is very less and the solute particles are very much more. और ये भी नोटिस किया होगा कि वो आचार में या पिकल में या जाम और जेली में उतना ज़्यादा फंगस नहीं लग पाते या वो कॉन्टामिनेट नहीं होते. यू नो व्हाई? वो हाइपरटोनिक सॉल्यूशन रहता है तो फंगस का सेल जब वहाँ पे ग्रो करता है तो फंगस का सेल प्लाज्मोलाइट हो जाता है, वो वाटर लूज कर देता है एंड वो मर जाता है. लेकिन अगर पानी ऐड हो गया अचार में या जाम में, कहीं से आउटसाइड से अगर वाटर ऐड हो गया तो एक फिर खराब हो जाता है. इट इज बिकॉज ऑफ प्लाज्मोलाइसिस ओनली. ओके. And also plasmolysis or plasmolytic method is used to determine osmotic potential or osmotic pressure of a cell. How it is used, the how plasmolytic method is used, we will see now. Okay, osmotic potential OP. इसको मैं समझा देता हूँ. One container, two container, three container, four container, five container. There are solution, sucrose solution. Suppose it is 1, 2, 3, 4, 5. 5 containers. Here it is 0.5 molar, 0.5 sucrose, 0.6 m 0.7 m, 0.8, 0.9। ठीक है, ये इसका कॉन्सेंट्रेशन है, सोल्यूट कॉन्सेंट्रेशन। हमारे पास एक सेल है, सेल को रेड में दिखा देते हैं। हमारे पास एक सेल है, तो इसमें सोल्यूट कॉन्सेंट्रेशन कितना है हमको निकालना है। तो हम यूज करेंगे, वी विल यूज द प्लाज्मोलिटिक मेथड, मीन्स वी विल enter or we will divide this cell and enter each part into different concentration of solution ऐड करेंगे। तो यहाँ पे आ गया, यहाँ पे आया, यहाँ पे आया, here also it came, here also it came. So suppose here the plasmolysis did not start, here also the plasmolysis did not start, but here you can observe the plasmolysis start or incipient plasmolysis you observe. Here incipient plasmolysis you observed in the cell. So this will be the concentration of the cell because the plasmolysis just started, right? So the solution just become a little hypertonic. Here the solution is hypotonic, means there was no flow. Here also no, but here plasmolysis started. So approximately equal to the concentration of cell, right? So the concentration of cell would be a little bit more than 0.7. See, same. Osmotic potential is a pressure that pulls water into a solution due to dissolved solute. More solutes, higher OP. Determine plasmolytic method. इसको बोला है plasmolytic method. What is the procedure? Plant tissue लिया गया, sugar solution में रख दिया गया of known concentration. Right. The sucrose solution are known concentration. And observe under microscope to find the solution that causes incipient plasmolysis, just beginning to shrink. OP of that sugar solution is nearly equal to OP of the cell sap. Osmotic potential or osmotic pressure of the cell sap. Use standard tables to find OP of sugar solution. फिर स्टैंडर्ड टेबल्स देख के हम शुगर का सॉल्यूशन पता कर लेते हैं. Now water relation in plant cell. What is diffusion pressure deficit? So diffusion pressure deficit. Deficit means lack or less. जो भी कमी है उसको डेफिसिट बोला जाता है. What is deficit here? The diffusion pressure, right? Between what? The difference between diffusion pressure, the difference of diffusion pressure between the solution and the solvent. In case here the solvent is water and solution जो भी plant cell के अंदर है cell sap, that is solution. So whatever there is difference between the diffusion pressure of like difference of diffusion pressure between the cell sap and the solvent, that is water, that is diffusion pressure deficit. Means that much water the cell sap can absorb or can take or can suck. That's why DPD is also known as suction pressure. So the definition of DPD would be the difference between diffusion pressure of pure water and that of a solution. Pure water is nothing but the solvent, as water, and it is the pulling force. How much a cell can pull water from the surrounding. It is the DPD of that cell. Okay, DPD is nothing but water deficit or suction pressure of the cell, and DPD is directly proportional to concentration of solute. If a system has more number of solute, means less water or less solvent, so it can suck more water, it can absorb more water, it means high DPD. high diffusion pressure deficit. Means higher the diffusion pressure deficit, more water it can pull. So high solute means high DPD, and high DPD means less solvent. Okay. So DPD is directly proportional to concentration of solute. Means if solute will be more, DPD will be more. And if we will dilute the solution, solute concentration will decrease, hence the DPD will also decrease. Greater absorb water. Okay. The pressure relationship plant cell. Pressure we have seen osmotic pressure, turgor pressure, and wall pressure. Diffusion pressure deficit. Diffusion pressure deficit also a pressure. So osmotic pressure, pressure due to solute pulling water in. Means osmotic pressure is the pressure uh, creates or generated due to the presence of solute in a solution. That is osmotic pressure. More will be the solute, more will be the pressure. Turgor pressure, pressure exerted by cell shape on cell wall. Wall pressure, the pressure exerted by cell wall against the turgor pressure. Right. So DPD is equal to osmotic pressure minus wall pressure. How? And wall pressure is equal to turgor pressure only. Like how much, if this is plant cell, this is cell wall, how much pressure it will exert here? The same pressure will exerted by cell wall. So here that's why wall pressure is equal to turgor pressure. Then how DPD is equal to osmotic pressure in water is minus wall pressure. Diffusion pressure deficit and osmotic pressure minus wall pressure. Diffusion pressure deficit also depend on the solute concentration, right? So it also depends on solute concentration. Anyone, if someone can tell, I'm waiting. See, what happened in a plant cell, if it is a plant cell or any system, solutes are present here. So the solute, more the solute, the entry of water will be more, right? Entry of water will be more. But what wall pressure does, wall pressure restrict the inflow of water by applying a pressure against the entry of water. If the water enter here, turgor pressure will increase. So the wall pressure, it will restrict the inflow of water, right? So when DPD is more, means solute concentration is more, water will enter. So wall pressure is also increase, but osmotic pressure will decrease. That's why DPD is equal to osmotic pressure minus wall pressure, because it will increase the entry of water and it restrict the entry of water. And DPD is the suction pressure, or how much water it can take. It will zero, when it will become zero, OP is become equal to WP, or it is turgor pressure. Understood? A bit complicated, but did you get it? Let's see. Lifting OP done. So in fully turgid cell, yes, it is confusing. What you did not understand, Uday Lakshmi? Okay. Did you understand diffusion pressure deficit? What is diffusion pressure deficit? Diffusion pressure deficit, did you understand? Here, solution and solute plus solvent. Here, it is only solvent. It is only solvent. Okay. So there should be difference in diffusion pressure, right? So this difference is only known as diffusion pressure deficit. This is only known as diffusion pressure deficit. And why this deficit is created? Because of presence of solute. Here in solution, solute is present. That's why it has a potential to absorb the water or take the water, right? But the intake of water will be opposed by wall pressure, right? When a cell take water, the turgor pressure increases. So there is the wall pressure also opposite. And how long a cell can take water? Until the dpd becomes 0, right? So dpd depends upon the solute concentration also and the wall pressure also. See, in some cell the walls are very rigid. In some cell the walls are very rigid, cell wall, and in some cell the wall are very flexible. So where the wall is rigid, the wall pressure will be more there. And no matter what is the solute concentration in that rigid cell wall cell, it will exert a pressure that the water only enters until it satisfies the diffusion pressure deficit. It will not depend on solute alone. So dpd depends on osmotic pressure and turgor pressure both, or wall pressure you can say, or wall pressure. Turgor pressure and wall pressure same only. Now it is clear. So when dpd equal to 0, now it is clear that dpd is equal to op minus tp or wp. So when dpd is equal to 0, that the cell cannot take any more water, then it is op minus tp. So here op minus tp becomes 0 if dpd equal to 0, that cell cannot take more water. So in this case op is equal to tp. And when dpd is equal to 0, in fully turgid cell, there is no place to take more water. So in case of turgid cell, dpd equal to 0. So in turgid cell, op osmotic pressure is equal to turgor pressure. But in plasmolysed cell, what will happen? In plasmolysed cell, here Tp is equal to 0, because plasmolysed cell will not exert, plasma cell like this, it cannot exert any turgor pressure. So Tp equal to 0. So here in this case, dpd is equal to osmotic pressure in plasmolysed cell. Got it? See, In fully turgid cell, Tp is equal to OP. Plasmolite cell, Tp is equal to 0. So dPd is equal to OP. And here OP is greater than Tp. It's okay. In normal cell, Tp is less than osmotic pressure. So dPd is greater than 0. Why in normal cell dPd is greater than 0? Because in normal cell there would be some solute. If solute is there, it has some tendency to absorb water. Okay. And how we will determine dPd? The same method, plasmolytic method that we saw to identify the osmotic pressure. But what we saw, it was the classical measure of water absorption, which is now replaced by the water potential. It is the present concept, water potential. Got it? In modern physiology, we use the term potential right now, the water potential, and it is the result of different potential like pressure potential, Turgor potential itself, pressure potential, osmotic potential. By combining all this, we determine the water potential, and the single term denotes the energy status of cell. Okay. So what is water potential? Osmotic relations. Osmosis means movement of water. So what is the relations of the movement? How the movement occur, and how it depends on the potential of water. Potential is free energy. The energy available to do any work is potential. Like how much capacity do you have? It means how much potential you have. The capacity is potential only. And what kind of capacity? To perform some work or any kind of work, it is potential. So it is the energy status of water, basically free energy that is available to perform a work. So water potential is the energy status of water in a system to carry out some work. Yes, sum of solute potential and pressure potential is water potential. Just not, but gravitational potential is also there, metric potential is also there, but gravitational potential and metric potential are considered negligible at the cell level. And in plant physiology, we study everything at the cell level. That's why the gravitational potential and the metric potential are zero. Metric potential means the imbibition part. Imbibition only the metric potential. For the adsorption of water on the surface of the colloids, that is imbibition. So both the things we take, or it is close to zero or negligible, that's why we take it zero. And water potential is equal to pressure potential plus osmotic potential. And now many of students get confused in the sign. It will be negative or positive. Solute potential will be negative or positive, or pressure potential will be negative or positive, right? That you will get for that you will get clarity now. Okay. Sum of osmotic potential and pressure potential. Here also I have written. It governs water movement in plants. Okay. According to the this potential only, that how much water potential means दोनों सेल के बीच वाटर पोटेंशियल में डिफरेंस कितना है. The difference cause the flow of water in between the cells. If there will be no difference in water potential, there will not be any flow. So for the movement of water, movement of any solute, there must be difference in water potential between the two system or two cells. What is the unit? Bar, megapascal. Okay, and the SI unit is Pascal. It is Newton per meter square. And conversion I have given. 1 bar is equal to 0.987 atmosphere, and 1 megapascal 10 bar equal to 9. You can see that only simple conversion is there. So here key definition like water potential, the free energy of water available to do the work, as I said, that determines the direction of water movement. Higher water potential to lower water potential. It is just talking about water, right? So how water will move from higher water potential to lower water potential. The direction of movement will be like this, like higher water to lower water potential. Okay, and what is osmotic potential? Osmotic potential is also called solute potential. Okay, and solute potential will depend on the amount of solute that is present in the solution or in the solvent or in the water. So if we add solute into the water, then water potential will increase or decrease. Means the free water or the free energy of water will decrease or increase. Suppose it is a container full of water, only pure water. Okay. And for pure water, water potential is equal to 0. We saw for pure water, water potential is 0. And if we add solute, yes, it will decrease. If we add solute into it, the water potential is decreased. It means the water potential becomes negative, right? So for every solution, if it is not pure water, water potential would be negative. Okay. And water potential equal to solute potential plus pressure potential. So now among these one will be negative and another one will be positive. Water potential is maximum 0 and then negative only. So always negative. Solute potential always negative. Why? Because when you add this, it becomes negative only. Pure water is 0. So if you add any solution, any solute, so the potential of the entire system would be negative. Hence the solute potential always decrease the water potential. If you add solute to a pure water, it would decrease the potential. It means the solute potential is always negative. Okay, it is always negative. So whenever in water potential you add is equal to solute potential plus pressure potential. So this is always negative because it always decrease the water potential. Addition of solute will always decrease the water potential. Sir, if pressure potential is more than solute, then water potential is positive, no sir. But it will not occur, no, because maximum water potential is 0. Pure water has the water potential of 0, gives energy, free energy, it consider, but theoretically it is possible, but practically not possible. Pressure potential cannot be more than solute potential. We'll see when we'll see no pressure potential. What is pressure potential or osmotic pressure? We'll see now. Theoretically, we can say if pressure potential would be more than solute potential, then the water potential will be positive. But practically, the maximum water potential is 0. And pure water has the maximum water potential. Okay, what is pressure potential now? The pressure exerted by cell wall on the cell sap. It is Turger pressure. Right. The Turger pressure or the pressure exerted by cell sap on the cell wall, it is called pressure potential. And pressure potential is always positive. Why? It is exerting in pressure, no, it is external pressure. It is applying. When water enters the soil, the like cell sap exert pressure on wall, and if it is more, the wall exert the same pressure on the cell sap. So it is the external pressure applied to the system due to the entry of water. That's why it is always positive. But in plasmolysed cell or where there is no Turger pressure, the pressure potential is 0. The pressure potential is nothing but the Turger pressure itself. And gravity potential, it is basically depend on height. Okay, if more will be the height, there will be some significant value of gravity potential. But at the cell level, cell are so close to each other, right? there will be no difference in height. The water will move like this, no? It will not move like this to this. So gravitational potential will not play any role in plant physiology. So here we consider as negligible. Now osmotic pressure. The pressure we apply to stop the osmosis is osmotic pressure. See, here there are two systems, semi-permeable membrane, two systems. Water is moving from here. So the pressure here we exert to stop this flow. Water is moving from A to B. A to B water is moving. So here if you apply a pressure to stop this flow, this pressure will be osmotic pressure. Understood? What is osmotic pressure? The pressure required to prevent the water entry by osmosis. It is related to solute potential but expressed as positive value. Means this flow will be dependent on concentration of solute, isn't it? But here the pressure is external, it will be positive and it is stopping the flow that is occurring due to difference in solute concentration. So the osmotic pressure is equal to minus solute potential. Solute potential is always negative and osmotic pressure is always positive because it is external pressure that we exert to stop the process of osmosis. Diffusion pressure deficit we saw. It is also called suction pressure. OP minus TP we saw already. General water potential formula is this much. This is solute potential, pressure potential, gravitational potential. Gravitational potential at cell level is negligible, 0. So the formula will be this much. In plasmolite cell, this you can correlate. I have already explained. In DPD, difference between DPD and water potential also I have already explained, so you can easily understood this. And now what is imbibition? Nothing but absorption of water on the surface. Solid particle, only it will absorb here on the surface. It will not enter or it will not cross inside the cell. They only absorb on the surface of cell or the surface of any substance. Okay, that is imbibition. Imbibition is the absorption of water by hydrophilic substance. Hydrophilic means water loving. Only that substance will absorb water, no, which is affinity for water. If like oil doesn't have any affinity for water. It will adsorb or absorb the water, no. But wood have the affinity for water, not all wood. Okay, so only hydrophilic substance will show imbibition, or imbibition will only occur in hydrophilic substance, or the substance which has a love for water without solute diffusion. And solute will not diffuse, only water will enter. On the surface, the solute will not diffuse, or from the surface, the solute will not go to solvent. Only the solvent will absorb by the substance on its surface. That is imbibition. And the imbibition pressure is now known as metric potential and contributes to water potential of the imbibant. Okay, and the imbibition pressure is driven by the affinity. How much will be the pressure or how much will be the metric potential? It is determined by the affinity or hydrophilic nature of the substance. Okay, it is the imbibition pressure would be decided by the love between water and the substance. How much they can stay together that will be the determination, or that will be the determining point for the imbibition pressure. And imbibition pressure is also known as metric potential. Imbibient, the substance which will absorb water, is known as imbibient. Okay, imbibed liquid, the solvent that will be absorbed by the imbibient. Imbibition pressure, the pressure created due to absorption of solvent on the substance, or on the surface of substance, is metric potential. Yes, in case of in plant, the metric potential is considered very less, negligible. That's why the water potential component is equal to solute potential plus pressure potential. But in case of soil, metric potential is not negligible. In soil, there is another water relation. It is just inside the plant. So in soil science, there you will be see that like a little difference is there when we will talk about water potential inside the soil. I am telling you the water potential inside plant. Okay, in case of plants, the metric potential is almost negligible. And metric potential is the modern term for imbibition pressure. Okay. And metric potential is always negative. Why always negative? Why? See, this is a substance in a system. It will absorb some free water. Only solvent it will absorb, right? Only solvent, no solute. So if it absorb the substance, then in the system, solute concentration will increase and water will decrease. So it will reduce the water potential, right? That's why it is negative, always negative. So pressure potential is always positive, solute potential is negative, and metric potential is also negative. Only pressure potential is positive. Water potential of ambient, see. Okay, so this is the end of the class. Do you have any doubt? You can ask if you have any doubt. And this is the last class on the YouTube. After this, there will be classes on paid class only in the app itself. Any doubt? Chat disconnected. Okay. Any doubt, student? So whatever your doubt will be, we'll clear in the paid classes itself. There we will get interactive classes. We can talk with each other. So it will be better to talk and solve the doubt. Here you are typing and I am explaining you. Okay. Are you enrolled for the course, Uday Lakshmi? Have you enrolled for the course? Okay, then actually I have also not seen the app and the features. Today I will check the app features and everything, and also I will talk with the admin to upload the notes and everything in app. Only class 1 is uploaded for plant physiology or tissue culture. Tissue culture only class 1 is uploaded in the app. The class only, no? It will be uploaded. By today 10 p.m. or by tomorrow morning, it will be uploaded. Don't worry. I will ask them to upload. It will be uploaded, and I think today or tomorrow we'll start the paid class as well. So we'll first start with tissue culture. We'll complete tissue culture first on the paid classes. Then we'll shift to plant physiology. Okay, so let's end the class. We'll meet in the paid class. Till then, take care. Bye bye.